Today we are going to discuss one of the major pathway of fatty acid oxidation, that is beta oxidation. As we all know, fatty acids are considered as the major energy reservoir of the living system. So after their consumption, they need to be oxidized to yield energy. There are different ways of fatty acid oxidation, but major of them are beta oxidation cycle. So today we will discuss about the beta oxidation. What is actually beta oxidation? Beta oxidation can be defined as the oxidation of fatty acid on the beta carbon atom with the sequential release of two carbon fragments in the form of acetyl-CoA. That is, in one cycle, two carbon fragments are removed in the form of acetyl-CoA. It occurs in the mitochondrial matrix of the eukaryote cells, and whereas in prokaryote, it occurs in cytosol itself. Beta oxidation can be divided into three stages. Number one, activation of fatty acid, then its transport into the mitochondria, then beta oxidation proper in the mitochondrial matrix. Now, activation of fatty acid. Activation of fatty acids occur in cytosol. The fatty acid to get oxidized, they must need to get activated before they are brought about the oxidation. The fatty acids without their activation cannot be brought inside the mitochondrial matrix. Once the fatty acids are activated outside, they are degraded by the process of beta oxidation. So this reaction can be divided into two steps. The reaction of fatty acids and ATP to form acyl AMP and pyrophosphate. As we can see in this reaction, fatty acid was combined with ATP to form acyl adenylate with the removal of pyrophosphate. Then this acyl adenylate react with coenzyme A to form acyl-CoA and AMP molecule is free. This acyl-CoA will get transported across the membrane. This reaction is catalyzed by the enzyme thiokinase or acyl-CoA synthase. Once the fatty acids are get activated, now they will transport to the inner mitochondrial membrane. As inner mitochondrial membrane is highly non-permeable to the fatty acid. So, fatty acyl-CoA, this formed cannot directly enter the matrix of the mitochondria. So, this step of reaction take place with the help of carrier proteins called carnitine. This reaction can be divided into four steps. Number one, at first the fatty acyl group is transferred to the carotene producing fatty acyl-CoA carnitine. This reaction is catalyzed by the enzyme carnitine acyl transferase 1. The carnitine acyl transferase is transferred to the mitochondrial matrix by a carrier protein, that is carnitine acyl translocase, that is found in the inner mitochondrial membrane with facilitated diffusion proceed this process further. Now, the enzyme named carnitine acyl transferase 2, which is found in the inner mitochondrial membrane, catalyze the acyl group from fatty acyl carnitine to CoA, yielding the acyl CoA, and this carnitine is get regenerated, and this will cross the membrane to take the another fatty acyl CoA. Now, the major is beta oxidation pathway. Beta oxidation pathway can be divided into four steps. Number one, oxidation, then hydration, then again oxidation, and then cleavage. Now, acyl-CoA undergoes dehydration by the FAD-dependent flavoenzyme called acyl-CoA dehydrogenase. A double bond is formed between alpha and beta carbon. Then, acyl-CoA hydratase bring about the hydration of the double bond to form beta hydrolyl acyl CoA. This beta hydroxy acyl CoA dehydrogenase catalyze the second oxidation and generate NADH, which will enter the electron transport chain for its oxidation to yield energy. The product formed is beta keto acyl CoA. Now, this beta acyl CoA will get cleaved with the help of enzyme thiolase, yielding acetyl CoA and carbon chain with less than 2 carbon atom. This acyl chain will again repeat the cycle to yield another molecule. This pathway will continue until all until all the carbon chain of the provided fatty acid is totally converted to acetyl CoA. Thank you.